Welcome to the deep dive. Today we're looking at something genuinely groundbreaking in cancer research. Imagine scientists taking out cancer cells using, well, basically just light. No chemotherapy, no major surgery needed. It sounds a bit like science fiction, but the research is solid. People are calling it a quantum leap.
Oh, absolutely. It's more than just, a small improvement. This is a whole different way of thinking about fighting cancer.
Mhm.
What's so exciting is how it sort of sidesteps the usual problems we face with traditional treatments. It's a fundamentally new angle.
Okay, let's get into that. So the science comes from teams at Rice, Texas A&M, and UT, and it hinges on these molecules called aminocyanine. What's special about them, and how do they manage to only stick to cancer cells? That selectivity seems crucial.
That's exactly right. Aminocyanine is a type of photochromic dye. It reacts to light. But the clever part is the engineering. They're built to latch onto specific markers you find mostly on cancer cells. Think of them like tiny, very precise homing devices. So once they've attached to the cancer cell, that's when the light comes in and does its work.
Right, they found their target. So what actually happens when you shine the light?
Okay, so once they're attached, they're activated using near-infrared light. And that type of light is key because it can go deep into tissue without causing much collateral damage. This light makes the aminocyanine molecules vibrate, but not just randomly vibrate. They vibrate incredibly fast and in sync, billions of them, essentially hammering away at the cancer cell's membrane at the same time. It's like, well, they call it molecular drilling or molecular tearing.
Molecular drilling.
Yeah, it generates this huge mechanical force that literally just rips the cell membrane apart. The cell basically ruptures and dies very quickly.
That sounds incredibly direct, physical destruction. So this molecular drilling, what results are they actually seeing in the lab? I heard the initial findings were pretty striking.
They really were. In the lab, just using cell cultures in petri dishes in vitro, they saw a 99% elimination rate for melanoma cells.
99%, that's huge.
It is. But of course, the big question is always, does it work in a living organism? So the next step was animal models in vivo. And in mice with tumors, this technique led to a complete cure in about half of them, around 50%.
50% cure rate in mice. That's very encouraging for this stage, isn't it?
Extremely encouraging, yes,
Yeah.
especially for a completely new approach like this.
And you mentioned this relies on physical force. How important is that, the fact it's not chemical? Because we always hear about drug resistance being such a massive hurdle in cancer treatment.
Right. That's perhaps the most revolutionary part. Think about it.
Yeah.
Cancer cells are masters of adaptation. They evolve resistance to chemotherapy drugs over time. It's like they learn to diffuse our chemical attacks. But how does a cell learn to resist being physically torn apart?
It can't really, can it?
Exactly. There's no biochemical pathway to block or adapt to. It's like trying to develop resistance to a hammer blow. Plus, as I mentioned, using near-infrared light is brilliant because it goes deep, and crucially, it causes very little damage to healthy surrounding tissue. That high selectivity means you potentially avoid all those awful side effects you get with chemo or radiation. It opens the door to truly non-invasive options.
So, with all this promise, where does the research go next? What are the immediate hurdles?
Well, the next phase is really about refinement and logistics. How do you get these molecules delivered efficiently to tumors deep inside the body? How do you scale up production? And then, of course, planning for human clinical trials. That's a very careful, step-by-step process. They'll likely start with specific types of solid tumors where delivering the light is more straightforward. It's definitely a journey, probably several years, but the potential payoff is pushing things forward quickly.
It really is amazing, the idea that simple light could become this incredibly precise, lethal weapon against cancer cells. It just shifts the perspective.
It absolutely does. For me, what's really profound is this idea of harnessing basic physics, fundamental forces at the molecular scale to treat disease. Imagine a future where we can tackle challenges like drug resistance, not with ever more complex drugs, but with something as fundamental as light, delivered with pinpoint accuracy. It's a powerful thought, isn't it? How sometimes the simplest tools, used in the right way, hold the most extraordinary potential.